Okay, so today's lecture, we're looking at glycolysis. So glycolysis is actually a term that consists of two words. So you have glyco and lysis. So we are saying you have the glyco on one part and the lysis. So the glyco refers to glucose, and the lysis refers to breakdown. So basically, this is a process, or this is a set of reactions that converts or break down glucose into two pyruvate molecules. So the whole process is interested to break down glucose into two pyruvate molecules. So it's a cascade of reactions, actually. So it's a cascade of reactions, or a set of reactions that results into the breakdown of glucose into two pyruvate molecules. It's very important for us to understand that. What is the aim of breaking down this glucose? So we are breaking down this glucose in order to produce ATP. And we know that this ATP is basically the fuel in cells. The fuel in cells. Other than producing ATP, we are interested to produce other intermediates. So these other intermediates are compounds that are going to be used in other biosynthetic pathways. Another aspect that we want to make mention here about glycolysis is that the whole process occurs in the cytosol of both prokaryotic and eukaryotic cells. The whole process is actually essentially anaerobic, meaning that it occurs in the absence of oxygen. So like how I said already, the role of glycolysis is basically to produce ATP and also other biosynthetic intermediates. We did make mention that glycolysis basically occurs in the cell cytosol of all tissues. Of all tissues of the body. So cells such as red blood cells, which are basically devoid of the mitochondria, depend entirely on glycolytic processes to produce, depend on glycolysis to produce ATP. because these do not have mitochondria. So my voice apparently is breaking. Let me just remove my headsets. Maybe it's okay. I hope now I'm clear. Just give me an indication with the text that I'm clear. My voice is no longer breaking. Okay, great. And we can move on. Also, contracting muscles. The process also occurs, that is the glycolytic pathway, occurs also in muscles. Sorry about that. So this occurs also in muscles. Due to the occlusion of blood vessels by muscular contraction, you'll find that here the concentration of oxygen goes down, and therefore, since glycolysis is largely an anaerobic process, you find that this process takes place, or rather takes precedence in the production of ATP. The other cells that utilize this process more prominently are the cornea, the lens, the kidney, cancer cells, and also the GIT. These basically produce ATP via the glycolytic process. Now, what is the biological importance of biological importance of glycolysis? So one, we are saying that this is the process that yields ATP. That is for the production of ATP, which is the fuel. It's the major energy source, like you mentioned earlier on, major energy source of certain tissues, which I mentioned earlier, like red blood cells, the cornea, the medulla of the kidney, and so on and so forth. It provides the, let's say, requirements, or it provides the necessary compounds for the process that occurs in the Krebs cycle, also known as the citric acid cycle. It is also a link in fat metabolism. So it's also a link in fat metabolism. The products of the glycolytic cycle, which is pyruvic acid, goes on to be decarboxylated to form acetyl-CoA, and the acetyl-CoA then goes into fat metabolism. It's also involved in the production of 2,3-diphosphoglycerate, which is important in tissue oxygenation. Within the notes that I'll send, you see the other functions. Now, the whole process, the whole glycolytic process, is thought of comprising of 3 stages. Okay. Please do not turn on your camera. We do not need to see you. So the whole glycolytic process is thought of as being comprised of 3 stages. Stage 1 is the conversion of glucose to fructose 1,6-bisphosphate. Stage 2 involves the breakdown or the cleavage of fructose 1,6-bisphosphate into 2,3-carbon into 3-carbon compounds, and stage 3 involves harvesting of ATP. So, harvesting of ATP. So these are the 3 stages that are involved in glycolysis: conversion of glucose into fructose 1,6-bisphosphate, breakdown of the produced compound into 2,3-carbon compounds, and also the harvesting of ATP. So the breakdown of glucose itself, which is the whole process of glycolysis, into 3-carbon pyruvate compound occurs in 10 steps. So the 10 steps, so let's say glycolysis involves 10 steps. And the 10 steps result in the production of pyruvate. So the pyruvate, sometimes they, because the pyruvate is the conjugate base of pyruvic acid. So sometimes in other texts, you will find they will use pyruvic acid. So the first 5 steps of the 10, these are referred to as the preparatory phase. So here, the preparatory stage is also referred to as the investment stage. So in the investment stage, 2 molecules of ATP are invested, or rather are used. 2 ATP molecules are used to cleave the hexose compound, which is sugar. The last 5, these constitute the payoff phase. So in the payoff phase, this is where now we are actually harvesting. This is where we are harvesting the ATP. So those are the 10 steps. Now, let's look at the individual 10 steps of glycolysis one by one. So let's start with step number 1. So step 1, which is reaction number 1, involves the phosphorylation of glucose. So the phosphorylation of glucose to form glucose 6-phosphate. To form glucose 6-phosphate. So you can write the word equation and then include the structure. Structure for us is very important. When you write these things, you need to show the structure. So we have glucose being converted to glucose 6-phosphate. So we are adding ATP. We are getting ATP plus. The enzyme that is involved is hexokinase. A kinase enzyme, kinases, these are enzymes that transfer a phosphoryl group from ATP to a receiving compound. So these are kinases. They transfer a phosphoryl group from ATP to another compound, in this case, glucose. So you can draw the pyranose ring, the glucopyranose, or you can draw the straight chain. So it is really up to you. So we can start with the pyranose ring, glucopyranose. So we have CH2OH. So that is the structure for glucose. And then we are saying we are putting in ATP and getting ATP plus, and the catalysis of hexokinase. And then we are getting glucose 6-phosphate. So the sixth carbon is the one which is phosphorylated. Two minus, like that. And then the rest of the structure comes back. So we have OH, 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 and OH. So here on the left, we have glucose. And this side, we have glucose 6-phosphate, which is produced. Uh-oh, I'm breaking again. Okay, so I hope I'm clear now anyway. Probably it's just network where you are. I hope that is not the case for everyone. Okay, so that is step number one. The phosphorylation of glucose to form glucose 6-phosphate. Let's go to step number two. So step number two is basically the isomerization of glucose 6-phosphate into fructose 6-phosphate. So step two is glucose 6-phosphate being converted to fructose 6-phosphate. The reaction is catalyzed by the enzyme phosphoglucoisomerase. So phosphoglucoisomerase. So this reaction even from the name of the enzyme, you can tell that this is just an isomerization reaction. Okay, so again, we write the structure for glucose 6-phosphate. So we have CH2, OPO3, 2 minus. Being acted upon, so this is a reversible reaction, being acted upon by phosphoglucoisomerase. So phosphoglucoisomerase, and then we are forming fructose 6-phosphate. That is 2 minus. There we have a CH2OH. We have an OH on top, and an OH down. So here, this is fructose 6-phosphate. And here I've got glucose 6-phosphate. Now, in a biochemical reaction, anytime you've got a reversible reaction, it means that this reaction can proceed in either way, depending on which compound needs to be formed at a particular time. But for step 1, you can see that the catalyzation of hexokinase is basically a non-reversible reaction. And this is because the reaction is a limiting reaction, or it is the rate determining reaction. So this one is the rate determining, meaning that when this step happens, then you are most likely going to get the product, which is pyruvic acid, which is at the end of the reaction. Okay, let's go to step number 3. So step 3 involves the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And this reaction, so 1,6-bisphosphate, this reaction is a phosphorylation reaction. So fructose 6-phosphate is phosphorylated. This means that an ATP molecule is added. So an ATP molecule is added. Okay. I really don't understand why people are saying they can't get me, whether it's network here or there. Okay, so I've got less than 8 minutes. Let me just finish, then we see what will happen. So we can draw the structure. So we have fructose 6-phosphate. So fructose 6-phosphate Plus ATP. Then you have ADP plus catalyzed by phosphofructokinase. This is also abbreviated as PFK. And then we are getting fructose 1,6-bisphosphate. Again, we can draw the structure. So we have OH2C2 minus. CH2OH. There we have an OH, OH, and OH. So this is fructose 6-phosphate. And then add to it ATP. Then we are getting ADP plus. This one is also a non-reversible reaction catalyzed by PFK. And then we have the product this side, OH2, 2 minus. CH2 or PO3, 2 minus. OH, OH there, OH like that. So this is fructose 1,6-bisphosphate. So that is step number 3. Let's end with the step number 4. So step number 4 is the cleavage of fructose 1,6-bisphosphate into 2 3-carbon molecules, namely glycerol, aldehyde 3-phosphate. Oh my goodness. Glycerol aldehyde 3-phosphate. And dehydroxy. So that's dehydroxy. So dehydroxy acetone. So dehydroxy acetone phosphate. So this is abbreviated as DHAP. And the glycerol aldehyde 3-phosphate is abbreviated as G3P. Okay. So we can go ahead and now just write the cleavage. So the whole process, this is catalyzed by the enzyme aldolase. So we can write the word and then write the structure. So we are saying fructose 1,6-bisphosphate. Maybe I can use a different color. So fructose 1,6-bisphosphate being cleaved. This is also a reversible reaction, being cleaved to form glycerol 3-phosphate plus dihydroxyacetone phosphate. So we can draw the structures, and we are saying that this is under the influence of aldolase. So we can draw the structure of fructose 1,6-bisphosphate. We're saying that is O3 PO H2C 2 minus. CH2 OPO3 2 minus. Then we can substitute OH, OH down and OH. So that is fructose 1,6-bisphosphate. And we are forming two compounds, glycerol 3-phosphate. So CH2 OPO3 2 minus. Let me write them in the same order I've written them. So we have, let me start with glycerol 3-phosphate so that we do not confuse things. So we have so this is a double bond, it's a carbonyl oxide, OH and H there. So this is glycerol 3-phosphate plus dihydroxyacetone phosphate. So it's CH2OPO3 2 minus. Like that. And I'm out of time. Okay, so we'll continue in the next session. But this is dihydroxyacetone phosphate. Okay, so I'll send a link, then we can proceed. Step 5 up to 10. Okay. Thank you.